So next you have the nucleus. So the nucleus is surrounded by a membrane itself called the nuclear envelope or nuclear membrane. Let me write in nucleus for you. So the envelope has two bilayers that separate out the nucleus from the cytoplasm. So that makes it kind of unique as far as organelles goes. So anyway, the nuclear envelope has pores in it. So they're basically little holes. And each of those pores allow things to go in or out of the nucleus. And it's going to regulate that. It's not going to just let anything in and out of the nucleus. But anyway, so as you know, DNA is located in the nucleus. So in order for the genetic code to get out, things have to happen, and we're going to do an entire unit on that later. But DNA has to make RNA, and then that RNA then leaves the nucleus through those pores to go to like the ribosomes to make proteins. So anyway, inside the nuclear envelope, you have the nuclear lamina, which is just a bunch of like protein filaments. It's kind of like a cytoskeleton for the nucleus, and so that gives that nucleus the little spherical shape. Kind of looks like a little ball. Anyway, so things that would be made inside of the nucleus are going to be RNA, proteins, and those things are going to be made from DNA because everything basically starts with DNA. So DNA makes RNA, and then that RNA can in turn make proteins. The structure shown above is found in what part of a eukaryotic cell? So hopefully you recalled from the biomolecules unit that this is a double helix, and so it is DNA. And in a eukaryotic cell, hopefully you recall that DNA is located in the nucleus. So this is a picture of a standard animal cell. So I just wanted to point out in the nucleus here, you see DNA, it kind of just looks like a bunch of squiggles. See all that? So anyway, I just wanted to show you that. You can also get a good view of the nuclear pores. So inside of the nucleus, you have the nucleolus. So the nucleolus is the little ball inside of the nucleus. It's a little structure inside of the nucleus. It's also called the little nucleus. So one thing that the nucleolus does is it stores the RNA that forms ribosomes. That's rRNA, ribosomal RNA. So anyway, a lot of people, they get the nucleolus mixed up with the nucleus. The nucleolus is inside of the nucleus. So this is a great picture of the nucleus because it shows your nucleolus here inside of the nucleus. I can't draw a circle very good clearly. And then you have your DNA. And DNA is in the form of chromatin here because this cell must not be in the process of actively dividing. So if you remember the two ways that you would see DNA, then you would recall that it can be X's, which is chromosomes, or it can be chromatin, which is how it's shown here. And I also like this picture because it shows you the nuclear pores so that the genetic instructions formed in DNA can leave the nucleus. Now, DNA doesn't actually leave the nucleus, but it does send a message out through messenger RNA. And then this picture also shows the nuclear envelope very well, and it shows that it's got a bilayer, a two-layered membrane. Another thing this picture does is it shows how the endoplasmic reticulum usually gets up close to the nucleus. And so you can see that the endoplasmic reticulum is right up close to the nucleus because eventually that mRNA is going to be formed in the nucleus. It's going to leave a nuclear pore and go out to a ribosome and form a protein. And then the endoplasmic reticulum is going to then transport that protein to another part of the cell. We'll be going over that in a few minutes. So then you have ribosomes. think y'all can see that one okay. So ribosomes are little dots, basically. So if you recall, when we looked at prokaryotic cells, one of the only things that was in it was cytoplasm, DNA, and ribosomes. So those were all the little dots. Ribosomes can be either just throughout the cytoplasm, or they can be on the rough ER, and that's what makes that the ER rough. There's a smooth ER, it lacks ribosomes. It does not have ribosomes. So ribosomes' function is that they are the site of protein synthesis. If you recall, synthesis means to make. Okay, so protein synthesis is making of proteins. Okay, so if you have any questions, there's lots of ways you could word this. You could say, what organelle is the site of protein synthesis? What organelle makes proteins? What organelle puts amino acids together to form a protein? Because remember, we learned in biomolecules unit that proteins are made from amino acids. Amino acids are the monomer, or the building block of proteins. So anyway, lots of ways you could word this. And again, ribosomes may either be in the cytoplasm or attached to the endoplasmic reticulum. You're going to see ribosomes in another unit later on. I've mentioned a couple of times that we're going to do an entire unit on DNA and RNA and protein synthesis. So we're actually going to do an entire unit talking about how protein synthesis occurs. So you'll hear more about ribosomes again at that point. So it's likely to see this on multiple tests in this class. So the endomembrane system, I touched on this when we were looking at the pictures, and we were noticing how the endoplasmic reticulum is going to be up close to the nucleus, and then the Golgi is going to be kind of behind it, and then you'll have maybe a little bit of space in the plasma membrane. So the endomembrane system is a system of membranes, and they're either directly like continuous or connected, or they have little vesicles is what they're called. And they're like little sacs, little circular bodies that carry something. They transport something from one place to another. So even though the ER and Golgi are not connected, they usually have a vesicle that takes a protein from the ER to the Golgi. So they're going to be kind of close together in the cell. Lysosomes and vacuoles are considered a type of vesicle. So anyway, organelles that assemble and transport proteins are going to include the ribosomes, the ER, endoplasmic reticulum, the Golgi, and vesicles. So again, this is between the nucleus and the cell membrane. And this is all like basically routing out what happens with the protein. And again, I told you that we're going to do an entire unit later on protein synthesis, but I want to just mention here, proteins are very important because proteins are how your genes are expressed on the outside. So if you think about it, when we talked about proteins, we said that they're a major structural component of many parts of your body. And in that, when your DNA, your genetic code, basically is to form all these different proteins. And when those proteins are made, your genes are expressed on the outside. And what I mean by that is maybe your gene for hair color is expressed. Maybe your gene for eye color is expressed on the outside, like where you can see it. When I say gene is expressed, you can see it. Maybe you had the BRCA1 gene for breast cancer your whole life in your DNA, and then when you were 43 years old, that gene was expressed because that was when the protein was finally made for that particular gene. But anyway, so the route of a protein and the movement of the protein through the cell is very important, and that's kind of why you'll notice that many of the organelles have a function that surrounds proteins. So anyway, typically, like a typical route for a protein, ribosomes make the proteins, and then they're transported to the ER. And in the ER, it's kind of like a highway. And then after it leaves the ER, it's packaged into a vesicle. So the vesicle is like a car, and it protects the protein from being just spit out into the cytoplasm. So the vesicle then usually takes it to the Golgi. The Golgi will change it, will modify it, and then repackage it into a new vesicle. And then that new vesicle takes it to another location. And sometimes that location might be out of the cell if it is waste or if the cell does not need it. So the building of proteins from amino acid occur on the cells. So remember we said the building of proteins, that's the same thing as saying like protein synthesis. And so that is going to be at your ribosomes. Peptide bonds between amino acids are formed primarily on the cells. So again, we're talking about peptide bonds being formed between amino acids. So that is the making of a protein. And again, that will be found, or that happens at the ribosomes. So which of the cellular structures labeled above is responsible for protein synthesis? Lots of questions on this, guys. So that's, of course, going to be your ribosomes again. So if you notice here, this picture is labeled the ribosomes on the rough ER. So here is a picture zoomed in of the rough ER. So if you look, it's showing our little ribosomes attached. And so it's very easy for the cell to make a protein and just spit it out from the ribosome on the rough ER into the ER. So it's made on the ribosome, in the ribosome, and then it gets spit out into the rough ER. So if you take a look at this picture over here, I like it. It's a little bit difficult to interpret for some people, but so if you look here, the nucleus, this would be the nucleus right here, and the nuclear membrane is actually connected. And so that mRNA is going to leave that nuclear membrane and go to one of these purple proteins. So all those purple dots are proteins. So those proteins will then make, the purple dots are ribosomes. So all those purple ribosomes are going to receive mRNA from the nucleus, and then that's their instructions to make the protein. And so the purple ribosome makes the protein, and then all the little green dots, those are proteins traveling through the endomembrane system. So it's just traveling through. Okay, and it's not a complete picture, but I think you can kind of get the gist of it. So now we have the endoplasmic reticulum. And of course, we usually just say ER for that. Way shorter. So, this is kind of the beginning of the endomembrane system. So it is a set of membranes, and it looks like a bunch of folded sacs or tunnels, and it's compared to a highway. So it's called the intra or inside the cell highway. So this is a path for things to be transported within the cell. Okay, so this is a transport mechanism. That would be a very key word for the endoplasmic reticulum is transport, or a synonym for transport. So a highway transports people. So watch out for that. It's probably talking about the endoplasmic reticulum, and it's going to usually say something about folded sacs or tunnels or something along those lines. So again, it's going to connect the cell membrane to the nuclear membrane. Or help connect it. And this is how you move things within a cell. So moving around within a cell, that kind of sounds like what transporting is. So what it's transporting is typically proteins. So again, the rough ER has the ribosomes attached to it. And so it's going to transport the proteins that were made on that ribosome. So it's going to go inside of the ER, and then travel to whatever other part of the ER, because it's a big kind of membrane system. It takes up a good portion of the cell. And so it could exit on the left or exit on the right, or, like taking different exits on the interstate. So wherever it decides to leave, a portion of the ER kind of wraps around the protein and encases it into a little sac called a vesicle. Now, be very careful because a vesicle also transports things. But a vesicle is going to be a small little membrane-bound sac versus a set of folded sacs or tunnels. Okay, so both vesicles and the ER transport things within the cell. But a vesicle is just a little tiny pouch, basically. So what happens is part of the ER actually kind of encloses that protein into a little pouch. I like to think of vesicles as like little cars, and then that vesicle is going to move to another part of the cell. So this keeps, the purpose of putting them into a vesicle is it keeps the protein separate from proteins that might be just produced by free ribosomes, or proteins that might come in the cell that are just floating in the cytoplasm that may be going somewhere else. So anyway, in order for proteins to be transported somewhere else, it needs to be enter the ER. So anyway, transport vesicles are going to bring them to the next step, and we're going to talk about the next step in just a second. But anyway, the inside of the ER, those all those tunnel-looking things are called cisternae. So again, the cisternae usually is going to be connected to the nuclear envelope. So you'll see the ER very close to the nucleus. And that's one way on a picture that you could distinguish between the ER and the Golgi. The ER is going to have those ribosomes attached to part of it, probably not all of it, because it will have a small section of smooth ER, but the ER usually is right up next to the nucleus, whereas the Golgi is usually closer to the cell membrane. So which of these structures below transports material within the cell? So transporting, again, I told y'all that was a key word for the ER. Notice vesicle is not a choice because those two are too close together. You would see a more of a description if you ever were given those two choices. But most of the time they don't put those two choices together. Okay, so again, two types of ER. You have rough and smooth. So rough, of course, has the ribosomes, and it transports the proteins. This is just something to know. Glycoproteins are proteins that have a carbohydrate attached to them. And glyco kind of sounds like glycogen, which is a polysaccharide, if you recall, that we store in our liver cells. Anyway, smooth ER does not have ribosomes, so that's why it's called smooth. And what it does is it makes lipids, like the phospholipids and steroids that make up part of the cell membrane. And different types of cells have different specializations of the smooth ER. One example is muscle cells have smooth ER that pumps additional calcium out. And it also stores them to release them when stimulated. Calcium is very important for muscle function. Gland cells that produce a lot of secretions. For example, the glands that produce insulin in your body. You're going to have more of those cells and more smooth ER in those cells. Sorry. Proteins must enter the endoplasmic reticulum to be transported to other parts of the cell, used in building new strands of RNA, synthesized into genetic codes, or excreted as waste. Hopefully you recalled transported was a key word there. So ER, transported. Okay. Thankfully this picture came across okay. So if you look up top, up here, we have the nucleus. Here, let me change colors. Okay, so we have the nucleus up here. So DNA forms mRNA in the nucleus. DNA is too big to leave the nucleus. mRNA takes the message out of the nucleus into the cell, into a ribosome. So if you see right here, the green ribosome, the red mRNA comes up to the green ribosome. And that green ribosome is going to make the protein. And so the protein, you see all these circles? That would be your protein. Okay, so your protein is going to then travel throughout the ER. And if you notice it traveled from the rough to the smooth. And then see the purple around it? So part of the ER goes around the protein and then it detaches here. It kind of pinches off and becomes a vesicle. So if you can see the purple around the vesicle. And then it gets transported to another part of the cell. Well, here we go. It's going to the Golgi. Okay, now the Golgi, we're fixing to get into Golgi. As you can see, my slides didn't come across because Apple and Microsoft don't totally connect. But anyway, so here we have the Golgi, and one of the functions of the Golgi is that it modifies. So modify means to change, basically. Okay, so it's going to change the protein somehow. So if you look here, we had two blues and two reds initially. 2 blues, 2 reds. But it kept the blues and it excreted the red as waste. This is the cell membrane, so it got rid of the red amino acids. Okay, so it modified and repackaged into a new vesicle and then excreted that as waste. Now, this new vesicle could go to a different part of the cell, possibly. So it repackages it. So packaging is going to be modified packaging is going to be something you see for Golgi. Okay, so now we have our Golgi. So you might see it called Golgi bodies, Golgi apparatus. I think I have apparatus. Golgi system, Golgi complex. It's still the Golgi. So again, that we looked at in the picture just a second ago, vesicles from the ER with those proteins in them are going to move through the cytoplasm to the Golgi. Golgi is called a stacks of flattened sacs or stacks of flattened membranes, and it's going to package and assemble different products in the cell. Usually that's going to be like proteins. There's 2 sites. The cis site is going to be closer to the ER, and so that brings the incoming vesicle into the Golgi. The other side is a trans side, and that's basically the opposite side, and it sends the vesicle out to its new location. It doesn't always exit the cell. So you might see the Golgi as seeing it as packaging and distribution center. Think kind of like Amazon. So it receives, it modifies, and repackages those protein. So then it can either store it for later, it can use it, or get rid of it, like we saw in the picture before. So anyway, those modified proteins, when they leave the Golgi, they are enclosed in a new vesicle that pinches off or buds off from the Golgi. So very similar to how it buds off from the ER. A lot of the times these vesicles might be lysosomes, which are an organelle we're going to be talking about later. So before the vesicles can exit the trans side, they are tagged with identifiers, like a phosphate group, and they're kind of like a mailing label or a zip code to tell it where to go in the cell. So again, Golgi's kind of like Amazon Prime. Christmas every day at my house. So anyway, inclusion bodies. So you have different inclusion bodies. One of those would be like a vacuole. These are organelles that have different fluids and materials that are brought in the cell, produced inside of the cell, or even things that are going to be leaving the cell. They're called inclusions or inclusion bodies. Okay, so here's a picture again. We have our ribosomes located on the ER, the rough ER, and the ribosome makes a protein. The protein then pinches off and becomes a vesicle. Goes to the Golgi to be modified, repackaged, and then we have the cis side here closest to the ER, and then the trans side here furthest from the ER. And then we have the new protein leaving in a new vesicle. So the next organelle we're going to talk about is lysosomes. So lysosomes are basically for digestion inside of the cell because they contain digestive enzymes. So they're like little sacs. So they look like little circles or spheres. And they're usually released by the Golgi. And so inside of them they have digestive enzymes that break down things. So they take care of foreign particles and also food and cell parts like cell organelles that are not functioning very well. So a foreign particle would be like something that's not supposed to be there. So anyway, so lysosomes are basically going to digest, or they're going to break down like food. If in a cell you have a massive rupture of a bunch of lysosomes, that destroys the cell. So this is called apoptosis. So this is like a programmed cell death. So normal cells, most of your so lysosomes are only going to be found usually in animal cells. Amoebas are animal-like protists. So they're a whole cell on their own. Anyway, so what was I saying? Okay, apoptosis. So what happens with apoptosis is the lysosomes, all the lysosomes in the cell basically rupture, and so they pretty much destroy the cell itself. So it's like an auto-digestion type thing. And the reason why this happens is because most of your cells are designed to only live a certain amount of time, with the exception of those cells that are amitotic that do not divide. So nerve cells are going to be amitotic, for example. So those are going to live, hopefully they live your whole life, obviously. And like heart muscle cells, you would hope that they would live your whole life. But things like skin cells or red blood cells or like white blood cells. So they should only last like a certain amount of time. And then once they get considered old for those cells, they're going to start to not work as good. And when your cells don't work as good, they could possibly become cancerous or just malfunction. And you don't want that. So those cells kill themselves because remember, cells are always going to be in a process of reproduction, except for, like I mentioned, those other cells before. So anyway, amoeba is an animal-like protist, and so they eat whole organisms. They're unicellular, so they're microscopic, but they're going to eat whole other unicellular things, smaller things, through phagocytosis. So phagocytosis is like cell eating, and your cells may also eat in this way, possibly. So anyway, when the amoeba eats something, it creates a food vacuole. Once it, think of like Pac-Man. Once it eats it, that's kind of like how phagocytosis is. Then it's called a food vacuole. And then what happens with that food vacuole, the lysosome will come up to it and combine with it, and then the lysosome helps digest the food. If you've ever seen anyone with webbed fingers or webbed toes, they didn't have as much lysosome digestion in the embryonic stage. So at one point, my baby would have had webbed hands and webbed feet and webbed neck. So one thing that is typical, my uncle has Down syndrome. So one thing that's typical with Down syndrome people is they might have some webbing in their hands and feet. So my uncle has just a little bit of extra webbing than what you would be considered normal in his hands. Now, it's probably not something you would notice, but if you're looking closely and if you're close to that person, then you would notice. So anyway, lysosomes are responsible for digesting that extra tissue. So here's a picture of, obviously, lysosomes. So this is showing here the food phagocytosis. The food goes in to the cell membrane, and then here's your food vacuole, and here is the food vacuole merging with the lysosome with the digestive enzyme, and then it digests it. Okay. hang on. And then it digests it, and then that's pretty much it. Another way that you could have happen is the lysosome could digest an organelle that's not working well. So that looks like a mitochondrion, and then you're left with parts of the organelle that can then be reused possibly by other parts of the cell. So again here, here's a lysosome, and it fuses with another part. Move that down. Okay. So peroxisomes are similar to lysosomes, so they also look like little circles, but they're more for detoxifying, okay, not necessarily digesting. Okay, so they also have enzymes. So these are similar in that they also use enzymes that are going to what they call oxidize harmful substances. Okay, so with the society we live in and the culture that we live in, we eat a lot of possibly harmful substances. So, like preservatives and different things. There's a lot of science out there looking at a lot of man-made foods, like think of like packaged foods. There's also science about like pesticides and weed spray, herbicides. And so some people have realized that those things build up and then they can become toxic and cause problems in your body. And of course, we know that alcohol, your body has to detoxify alcohol if you intake alcohol. Anyway, so peroxisomes are going to help detoxify from harmful things, gets rid of harmful things. Then you have endosomes. So endosomes are going to be where like cell parts go through, or like when you take in that food vacuole, where they go before it's digested by the lysosomes. So these endosomes are going to help sort to tell what is usable for that cell. What does that cell need? So it kind of makes like a keep and a discard type stack before it gets digested by the lysosome. That we don't want to digest anything that is usable. So here on the left, you would have your peroxisomes, and then here on the right, you're going to have an sorry, my iPad fell. Endosome. That would be, see this incoming food vacuole, and then it becomes the vesicle, and that endosome is going to help sort it out, sort what is needed. So that brings us to phagosomes. So this is another type of vesicle, and you can think of phago as eating. So this is cell eating or taking large things into the cell. So phagocytic cells, they're going to take in like a food particle, for example, and then it's going to be digested by the lysosome, or it could be detoxified by a peroxisome, or an endosome could look at it and sort out what will be needed for the metabolic, for the cell, for the metabolic energy in the cell. Then you have penosomes. So peno would be cell drinking. So these are also vesicles. So all these things look very similar. They look like little circles. So this is going to be drinking. So it's going to be like water and solutes, like, salt water, for example. So this would be the cell taking in like a large pouch of water. Then you have exosomes. So exosomes are also little vesicles. So it still looks like a little circle. They all kind of look the same. And they are vacuoles, so they store things, and eventually they are going to be expelled from the cell. Okay, so usually this is going to be things the cell does not need anymore or waste. So they are going to be expelled. So exosomes are things exiting the cell. Okay, so if you look here, we have something exiting the cell. So that would be a exosome. Okay, other organelles, mitochondria. So mitochondria is the plural, mitochondrion is the singular. Mitochondria are going to produce energy. Now, cellular energy or molecular energy in a cell is going to be called ATP, adenosine triphosphate. You might have remember learning briefly mentioning this in the biomolecules unit. So anyway, because it's producing energy, this is why we're calling it the powerhouse of the cell. So how your cells produce energy is through cellular respiration. Okay? Now, it's very important for you to know that every cell does this, plant or animal. Okay? So aero means air or oxygen specifically. So this is basically how you get energy. You take in glucose from food and oxygen and create ATP from it. And like carbon dioxide and water are byproducts. And you're probably a little bit familiar with that. So this is where breathing comes. So you have to breathe and eat in order to form energy. And this happens in the mitochondria in the cell. So that's why it's called respiration, because you're breathing to get energy, but you also have to eat too. So cells that have need a lot more energy, like muscle cells, are going to have way more mitochondria. That kind of should make sense, because muscle cells are going to use a lot more energy than other cells. So the mitochondria are distinctive because they have an outer membrane and an inner membrane. And the inside have cristae. So they're kind of, I think they're kind of like bean shaped or oval shaped. And it kind of looks like this. Those are the cristae, the little squiggles. And so you have what they call an intermembrane space. I told you the cristae. Okay, so they have the intermembrane space. So this is between the two membranes. You would have like a second membrane here. That's not going to be that big, but I'm not a good artist. So this would be the space, the intermembrane space right there. Anyway, so it's called the mitochondrial matrix. So anyway, this space is fluid filled, and mitochondria are unique because they actually have their own DNA, and they also have ribosomes and enzymes. So there's some theories of evolution that believe that prokaryotic cells ate or engulfed or phagocytosed another cell, and that other cell might have been mitochondria, and that's why mitochondria have their own DNA. So there is a theory out there that talks about that. And we'll learn about that when we get to evolution. Endoplasmic reticulum is to mitochondrion as road is to. Now this is the first time we've seen one of these questions, so I want to kind of just show you how to do it. So first off, I notice I have two organelles here. Okay, and so I need to figure out what is it asking for. So it told me road. So which one does road go with, the ER or the mitochondrion? Okay, so hopefully you figured out road goes with mitochondrion. So those two go together. So I'm looking for what goes with mitochondrion. Would control center go with mitochondrion, generator, storage tank, or toll gate? So hopefully you would pick generator. Okay, so these two go together, and it wants to know what goes with this. Okay, the control center would be like the nucleus. The toll gate would be like the cell membrane. The storage tank would be like a vacuole, which we're going to talk more in depth about in a few minutes. So this is a couple of pictures of mitochondrion. So if you see, they have kind of a distinctive appearance with the cristae and the double membrane. And this is just a cool picture. So again, we're in both plant and animal cells have these organelles. So I told you we were going to talk about vacuoles, and here we are. So vacuoles, it depends on what kind of cell you're in, what they look like. They look very different for plant cells versus animal cells, but they do both have them. So, like in an animal cell, they're going to be very small and they're going to be fluid filled structures, but their main function is storage. They store food, water, enzymes, lots of things. Okay, so they store stuff. Now, a centralized vacuole is going to be more for like a plant cell. But anyway, they might. This is all the things that they could do. They could stockpile or store proteins, other ions. They can possibly dispose of byproducts from respiration, the metabolic byproducts. They can hold pigments, and that's specifically going to be more for plant cells. And they might have defensive compounds. So think like poison ivy. That's a defense compound that's in the plant cells. So this is going to be more like plant, and this is going to be more like animal. So anyway, you got three main types. You got food vacuoles, so that's formed by phagocytosis, so basically cell eating. And then they eventually fuse with the lysosomes, which then digest that food. Then you have contractile vacuoles. So those are going to be in protists. Protists are typically like unicellular microscopic organisms that live in fresh water. So like ditches and ponds and lakes and rivers. And they pump extra water out. Because these cells live in water, they could easily burst if they got too much water in them. So contractile vacuoles kind of have a sun-like appearance. So they kind of look like that. And this would be like when they're not full. So anyway, it's still going to store water and things like that, but it will squeeze and contract to get rid of extra water. And then you might have a large central vacuole in plant cells. Now, in plant cells, you're going to have what's called the tonoplast, and that's going to surround that large centralized vacuole. So if you take a look here, notice the plant cell, the giant vacuole in the center, and then in the animal cell, you have multiple smaller vacuoles. So both have them, but they just look very different depending on the cell. And then here, let me move this. Here is that contractile vacuole that I was telling you about that protists can have. And you can tell this is a microscope picture. So this is a microscopic organism. And then if you look, we've got the food vacuole here. This is just showing the process. So 1, 2, 3, 4. So here's your food vacuole coming in. Food particle going in, forming the food vacuole. The lysosomes coming up and then digesting. So again, there's another one like this. Golgi apparatus is to vacuole as packaging is to. Okay, so what did they go with here? What does packaging go with? Well, hopefully you knew packaging goes with Golgi. And we're looking for what does vacuole go with. Okay, is it protecting, storing, absorbing, or hydrating? Well, your key word for vacuole should be storing. So now, the title of this slide, move it where you can see it, says found in animal cells only. These are only in animal cells. I've mentioned a few as we go along here. But anyway, one example is centrioles. Now, centrioles are found in pairs and near the nucleus. Notice this nucleus here, and you got your centrioles very close to the nucleus, and they're paired off. I think centrioles kind of look like a bundle of straws, but they're considered cylinder structures. And if you consider a straw as kind of like a cylinder. So anyway, what centrioles do is they're involved in cell division. Okay, so cell division is going to be like mitosis and meiosis. Okay, depending on what kind of cell we're talking about. So anyway, peroxisomes are going to have enzymes that transfer hydrogen and other things to oxygen, and hydrogen peroxide is poisonous, so that can be converted from a poisonous substance to a non-poisonous substance, water and oxygen. And that's just something really that is on the wrong slide. That would need to go back when we were talking about peroxisomes. Sorry about that. I'm editing things, and it's very easy to make mistakes when you're moving things all around and adding things in. I'm a human, sorry. Anyway, so here is some pictures. Okay, so this is just a up-close picture of centrioles. And so notice how they look like little pairs of tube-like structures. And then your centrioles in this picture are going to be here. I'll just draw a circle for them, but that's where they would go. And so in cell division, this DNA, the purple, the dark purple would be DNA, and it would have been lined up in the middle, and those were X's, they were chromosomes. And then this is actually called anaphase of mitosis, and so what happens is you'll notice that that DNA is moving to opposite sides of the cell, and eventually that one cell will split into two. So these centrioles help pull. They're spindle fibers. All these things are spindle fibers. All that red stuff, those are all spindle fibers. And so they're attached to the chromosomes. They anchor the chromosomes to the centriole, and it kind of reels them in like fish, like you're reeling in a fish. So it's pulling the chromosomes to the opposite sides. So eventually you can make your new cell, two new cells from one cell. Okay, so notice the title of this slide is found in plant cells only. Now, just to recall, remember I said lysosomes and cytoskeleton are also only in animal cells. So don't forget those because we mentioned those before. So anyway, plant cells only. Plastids. So plastids are only in plants. There's a couple of different types of plastids. So what plastids do is they make food and they also store food. So there's three main types of plastids. Now, the most common type that we think about in plants, because plants are known for their green color, is chloroplast. So chloroplast, chloro literally means green, if you recall from your prefixes. Okay, so chloroplasts are going to be green, and they are responsible for green parts of the plant. So the main function of chloroplast is photosynthesis, and this is how plants make their own food. So they trap the light energy for photosynthesis to happen. So then the next type is called myeloplast, or another name for them is leucoplast. Leuco means white, if you recall. So these are a colorless plastid. So if you think about potatoes, the white part of the potato is going to be it's going to have leucoplast in it. And potatoes are considered like a tuber or root type vegetable, so they're going to store starch. So those leucoplasts are going to have starch, and remember, starch is hundreds of glucose molecules linked together, so that's why they taste so good. Anyway, then you have chromoplast. So chromoplasts are going to have different colored pigments. So they're going to have different colors there. So you pretty much have chloroplasts that are green, myeloplasts that are white, and then all the other colors you may see in a plant are going to be chromoplasts. That's the pigment. Okay, so, fruits can be lots of different colors. Bananas are yellow, oranges are orange, strawberries are red, blueberries are bluish purple. So, you have all these different colors, all the rainbow of colors that you could have in fruits and vegetables and flowers too. This is any plant, okay? So chromoplast, all the other colors would come from chromoplast. So anyway, then you have the stroma. So the stroma is the inside membrane, and it is going to be the part, it's got fluid in it, and it's going to have these thylakoids. So this is going to be usually inside of the chloroplast or these plastids. So it has thylakoids in it, and a stack of thylakoids is called a grana. This is going to be important, especially when we get to photosynthesis later on in the next unit. So this is a picture of a chloroplast. Okay, so again, I was mentioning to you the stroma and the thylakoid, and the granum is one stack of thylakoids. So you see multiple grana here. I like to think as one thylakoid as one lifesaver, or one pancake, but the granum is a stack of pancakes or a stack of thylakoids. So anyway, this would be where photosynthesis happens in plants. So then here you have myeloplast, and then you have your chromoplast. So on the left you have your myeloplast, and on the right you have your chromoplast. So another thing that plant cells have is a cell wall, and this is compared to animal cells. So plant cells have a cell wall. Now, of course, we mentioned that bacteria might have a cell wall, and we also mentioned fungi have a cell wall. So don't forget, plants are not the only thing with a cell wall, but animals do not have a cell wall. So cell walls are made up of a mixture of proteins and carbohydrates, and in plants they will have the polysaccharide cellulose. In fungi they will have chitin, which is actually a polysaccharide that has nitrogen in it. So anyway, remember, a polysaccharide is a complex sugar or a mini sugar. Okay, so where the cell wall is outside of the cell membrane in plants, and its purpose is strength and rigidity. You can think of rigidity as like a firmness. It does not affect things going in and out of the cell. So it does not affect passage of substances in and out of the cell. That is the cell membrane's function, always. Just because you have a cell wall doesn't change the function of the cell membrane. The cell membrane's function stays the same all the time, okay? So the cell wall just gives it an extra strength and an extra firmness to the cell. Depending on how thick it is and what chemicals are in it, depends on the species of plant. And so it's going to have the primary cell wall, then a middle part that's called the lamella, that has polysaccharides in it. And you probably already know that sugars are kind of sticky, so it's going to have the polysaccharides in it. Then it's going to help it stick to the other layer, the secondary cell wall. Okay, so here's a picture of a plant cell. And you can see the kind of beige yellow. Here, let me change colors. The kind of beige yellow part here would be the cell membrane. And then the green would be your cell wall. And then, of course, I know this is a plant cell also because it's got a large central vacuole and chloroplast. And notice the chloroplasts look a little different from mitochondria. Now, I'm going to take this opportunity to tell you a little bit more about chloroplasts. Chloroplasts are similar to mitochondria because they also contain their own DNA. And so, again, back to one of those theories of evolution that we'll talk about later on in the semester, it is theorized that chloroplasts were also eaten by a prokaryotic cell. And so they think that the first prokaryotic cells turned into eukaryotic cells by eating these other prokaryotic cells that had their own DNA. And so that's why they think chloroplasts and mitochondria have their own DNA, because they think they were eaten originally by a prokaryotic cell. And then after the prokaryotic cell ate them, it became eukaryotic eventually after forming a nucleus. And it got all the other organelles. That's the theory anyway. Which of the following organelles are not in both animal and plant cells? Mitochondria, cell membrane, chloroplast, or vacuoles? Hopefully this was easy for you. Remember, all cells have a cell membrane. Okay, and both plant and animal cells will have vacuoles, but in plants one will be larger than the others. And all eukaryotic cells, which are plant and animal, are going to have mitochondria. But only plants will have chloroplasts because you are not green. When viewed under a microscope, a cell has several structures visible, including a membrane-bound nucleus, a cell wall, and several mitochondria. What type of cell is it? Well, because it has a nucleus, I can automatically eliminate prokaryotic and bacteria, because prokaryotic and bacteria are the same thing. So when I automatically look, does it have a nucleus? And so that tells you if it's prokaryotic or eukaryotic. So it has a nucleus, so I know it's eukaryotic. Now, if it has a cell wall, then hopefully you would pick plant cell, because animal cells do not. So all the following statements about plant and animal cells are accurate except, so you're looking for the inaccurate statement or false statement about plant and animal cells. Plant cells have a cell wall, but animal cells do not. That is true. Animal cells have mitochondria, but plant cells do not. Mitochondria? Yeah, plants need that too. So this is going to be your false statement, but let's look at the others. Animal cells have centrioles, but plant cells do not. That is true. And both plant and animal cells are eukaryotic. That is true. They both have a nucleus. When viewed through a microscope, which of the following would indicate the sample contained eukaryotic cells? Okay, so remember, eukaryotic, EU, and karyo. Y'all remember what they stand for? U was true, karyo was nucleus. So, eukaryotic has nucleus. The cell shown above is not an animal cell because it has a membrane-bound nucleus. No, animal cells have it. It has a cell wall. Right, animal cells do not have a cell wall, and this cell has a cell wall. Animal cells can also have mitochondria, and the size does not matter. So, that brings us to some kingdoms. So I wanted to introduce to you the six kingdoms, because we did talk about several of these as we were going through this section. So all living organisms, if you are alive, if you're a living organism, you have to be grouped into one of these 6 kingdoms. And they're grouped based on similarities. So 2 of the kingdoms are prokaryotic, and the other 4 are going to be eukaryotic. And I mentioned this earlier in this section of notes. So Archaebacteria and Eubacteria are going to be considered prokaryotes. The others, fungi, plant, animal, and protists, are going to be eukaryotes. So remember, eukaryotes have a nucleus, and prokaryotes have no nucleus. So we're going to talk about each one of these kingdoms now. So this up here should say prokaryotic kingdoms. So the first one is Archaebacteria. So Archaebacteria, you need to think as like archaic. And when I think of something that's archaic, I think very old. Okay, so archaic bacteria are going to be prokaryotic. And all bacteria are also unicellular, so you'll see they have that in common. And they do resemble typical bacteria, but they are a little different. But Archaebacteria are considered the oldest living things. They are very, very, very old. And one of the reasons why they're so old is they're able to survive extreme conditions. So they're also known as extremophiles. Okay, so they can usually live super duper hot conditions like the inside of a volcano or hot springs, or super salty conditions like a salt lake, because normally living things would not survive in those conditions. So then the second kingdom, kingdom number 2, is Eubacteria. Remember, Eu means true. So these are what we consider true bacteria. So these are going to be your more everyday type bacteria that you're exposed to. Again, they're going to be prokaryotic and unicellular, because all bacteria are going to have that in common. But Eubacteria are going to be things like strep throat bacteria, Streptococcus aureus, or like pneumonia bacteria, or different things that we're exposed to more on an everyday basis. Okay, so this slide should be titled Eukaryotic Kingdoms. So our prokaryotic kingdoms we just briefly talked about. Now we're going to briefly talk about our eukaryotic kingdoms. So remember, eukaryotic means it has a nucleus, but it also has organelles. Okay, so some of the organelles are going to be the mitochondria and the chloroplast. And they need to be able to get energy, and those are the 2 organelles we use to get energy. Now, of course, I said we, but plants use chloroplast. We don't have those. So anyway, photosynthesis again is going to occur in the chloroplast in plants, but there's also plant-like protists. And plant-like protists may also, they're plant-like because they go through photosynthesis. So algae is an example of a plant-like protist, and algae may be green. There's some other colors of algae too, but that's just an example. Algae is not actually a plant, it's a protist. So anyway, photosynthesis turns light energy, water, and carbon dioxide into oxygen and glucose. Now, in cellular respiration, and this is going to be all living things are going to go through cell respiration. This is all living eukaryotics. This is going to happen in the mitochondria. So plants do this too. So plants make sugar, they make glucose, and then when they need to break it down for energy, this happens in the mitochondria. This is also how you get energy. So you use food, glucose, and oxygen when you breathe. That's what's called respiration. So breathing. So you're breathing in oxygen, giving off carbon dioxide. So you're breathing in oxygen and taking in glucose from your food to release energy. Carbon dioxide and water is another byproduct not listed. So anyway, fungi, plants, animals, and certain types of protists go through cell respiration. So again, we're in the eukaryotic kingdoms. So the third kingdom and the first eukaryotic kingdom we're going to talk about is protist. Now, kingdom Protista is also known, I'm not writing, the catch-all kingdom. So basically, if it doesn't go in the plant, quite fit in the plant kingdom, then it might be a plant-like protist and go in the protist kingdom. If it doesn't quite fit in the animal kingdom, it might be an animal-like protist. And it doesn't quite fit in the fungi kingdom, then it might be a fungus-like protist. So these protists are going to be eukaryotic, so they're all going to have a nucleus, but they're usually unicellular, and unicellular typically means microscopic. Now, if you get a bunch of them together, of course, you'll be able to see it. Now, because it's a catch-all kingdom, and because you have some that are plant-like and animal-like, they may be autotrophs or heterotrophs. So they might make their own food like a plant, or they might eat like we do. Lots of examples. Again, this is called the catch-all kingdom. So this is going to be your most diverse out of all of your 6 kingdoms. And diverse or diversity means differences. So the organisms in this kingdom are going to have the most differences when you compare it to all the other kingdoms. So you have algae, paramecia, euglena, protozoa, slime molds, and of course the amoeba, which we've mentioned multiple times now. One thing that you'll notice about protists is that they typically live in fresh water. So they have that contractile vacuole that we mentioned before. And so in our next PowerPoint for this section, we're going to talk about hypotonic solutions, which could cause the cell to burst. And so a contractile vacuole is to help prevent that and to pump excess water out. Now, protists, because they're unicellular, they usually have some form of locomotion. They might have cilia, they might have flagella, they might even have a pseudopod like an amoeba. And we've mentioned pseudopods before earlier as well. So then you have fungi, kingdom fungi. Now, fungi are mostly multicellular. Of course, you have yeast, like yeast we use to make bread is actually fungi, and it's unicellular. But most every other fungi is going to be multicellular. So, with fungi, they are eukaryotic again because we're in the eukaryotic kingdom. So they have a nucleus and they have all those organelles. But fungi are heterotrophs. They do not make their own food. So they are going to go through cellular respiration just like a human would, but they eat differently. They use what's called extracellular digestion. So basically what happens is extracellular digestion would be they digest the food first, then take it into their cells. And they do that through something called hyphae. I like to think of hyphae as like if here's my mushroom, that's weird, but anyway, the hyphae are kind of like little tentacles that come out, and they can't see them. They're usually microscopic, but they go out into the surrounding area and they release the digestive enzymes to digest it. They're breaking down usually like organic material, like dead leaves, dead worms or whatever. Anyway, so they release digestive enzymes, and once the food is digested enough to be small enough to cross into the fungi cell membrane, then it can absorb the nutrients in the food. So that's called extracellular digestion. So fungi would include mold, mushrooms, and again, like I mentioned, yeast. So then we have plants and animals. So we kind of know a lot about plants and animals already, I feel like. So plants and animals are both going to be eukaryotic. They're both going to be multicellular. But all plants are going to be autotrophs, so they're going to make their own food through photosynthesis. All animals are going to be heterotrophs, so they must eat. So examples of plants are going to be, of course, vegetables, trees, grass, moss. Now, animals, some people might not realize that sea sponges are actually animals. Jellyfish, worms, any insect, anything, fish, reptiles, amphibians, birds, and of course, mammals. So this should say all of the following kingdoms include autotrophic except. So what does not include autotrophic animals? Hopefully you picked Animalia. Sorry, I told you the answer. So this should say which of the following is considered a living organism. If you remember, it is not. Sorry, it's not considered living. If you remember, we talked about a paramecia, which was a protist, a mushroom, which was a fungus, and bacteria, which it could be nitrogen-fixing bacteria is probably going to be eubacteria, but it didn't specify, so we don't fully know, but it's probably eubacteria. Regardless, we know bacteria is living, and we know viruses are not living. Okay, so this just says, name that kingdom. Okay, so which kingdom is going to have unicellular prokaryotic that live in both aquatic or terrestrial conditions? They cause disease and they're used in food and medicine. So this is basically like normal bacteria. So this is going to be eubacteria. I know it's bacteria because it's prokaryotic. And because they cause disease and are used in foods and medicines, I know that's my normal true bacteria. Again, name that kingdom. Okay, so it's going to include examples that are algae, slime molds, flagellates, ciliates, sporozoans, and amoebas. And I think we mentioned a couple of times what this one was. So that should be kingdom Protista or protists. Okay, so the kingdom that is unicellular prokaryotes and harsh environments. Remember, prokaryotes are going to be bacteria. Harsh environment, think ancient or old. So that's going to be archaea. Bacteria. There might be another A there. Can't remember. Examples of this kingdom are going to be mushrooms, mold, puffballs, mildew, and yeast. Hopefully you know that mushrooms are going to be in the fungi kingdom. Ancient prokaryotes, that's going to be archaebacteria. Okay, now this should say which of the following distinguishes organisms in kingdom fungi from other eukaryotic organisms. So what makes fungi special, basically? Is it that they're unicellular? Well, that's not true. Is it that they reproduce sexually? We didn't get into that, did we? Obtain nutrients by absorption through extracellular digestion? There we go. And remember, only that plants, they do not make their own food. Plants do that, not fungi. Okay, name that kingdom. Most are unicellular, but it's eukaryotic, and they may live in aquatic or moist habitats, so typically water, and they may be hetero or autotrophs. So hopefully you picked protista. True prokaryotes. Hopefully you remember true stands for you, and prokaryotes, the only example of prokaryotes is bacteria. Okay, last one. Name that kingdom. Eukaryotes, multi and unicellular, and they absorb nutrients as decomposers of organic matter. So we didn't mention this, but I wanted to put it here. Decomposers, eukaryotic decomposers, are going to usually be fungi. Now, some protists can be decomposers, but protists are mostly going to be unicellular. So, you'll need to look out for the next PowerPoint that is going to be on how cells transport things in and out.